our next speaker, and everyone can probably see a thread of care throughout each of these presentations, from the technical privacy and security from Rebecca Herold on IoT devices in the smart home, to how technology is being used and developed by manufacturers for the digital world in Rhys Farthing's talk, and now to the politics of care and control in another topic, synthetic biology. And it's my joy to introduce Emma Frow from the Society Policy Engineering Collective Research Group. We've got a lot of things to celebrate with respect to Emma. Emma's a joint appointment in the School of Biological and Health Systems Engineering and also the School for the Future of Innovation in Society. And she's recently won an early career research award titled Reconfiguring Life, Care and Control in Bioengineering. You can find some of Emma's videos and discussions and her conference presentations online. They're very informative. This one was titled Synthetic Biology in a Societal Context, done a very long while ago, but still holds its stealth as I've watched it at least twice now. So to the papers as well, please feel free to engage in responsible innovation research. Here we are. Here's another one of those topics which we've been talking about today. But please do go and download Emma's work, and the joy again of having the three of you as guests, but I'll stop sharing and let Emma take the floor.
Thank you, Katina, for that introduction. And thank you, Rebecca and Rhys, for two really interesting, scary, provocative talks this morning. And actually, I think there are some interesting connections between our projects. In particular, I think hopefully today I can share a little bit about maybe some of the more upstream work that seems to be happening in this field of synthetic biology. So what I'm going to do today is take you into the world of high-tech biotech and synthetic biology. Can you see my screen okay? I can't see any of you now, unfortunately.
Not yet. Not yet.
Oh, hold on. Let me see here.
There we go.
Okay, great.
Thank you.
You'd think by now I would be better at this.
You're an expert. I just know Zoom's picking up on all of us.
All right, so yeah, I started my career as a bench scientist, but I sort of quickly realized over the, during my time at the lab bench that actually the questions about science and technology that I was most interested in weren't the technical details of how to design my next experiment, but some of these bigger picture questions about the politics and the trajectories of science and technology. So after training as a bioscientist, I retrained in a field called science and technology studies, which I don't know how many of you might have heard of this, but it's a very kind of heterogeneous field that really tries to explore science and technology from all kinds of different disciplinary perspectives. So it's a pretty heterogeneous. If I had to describe the kinds of specific approaches that I take within STS, I'd say that I'm maybe part anthropologist, part sociologist with the kind of work that I do. So I'll try and give you a little bit of a flavor of that today. But it was when I was first sort of doing this retraining in STS after leaving the lab bench, this is a while ago now, 14 years ago, that I first dipped a toe into this world of synthetic biology. So it was 2007. I was based in the UK at the time at the University of Edinburgh. And the field of synthetic biology had been at the time starting to emerge in the US, and it was just beginning to find some traction in the UK. So again, I don't know with this group how many of you are familiar with synthetic biology, so I'll just take a minute and give you, a very quick snapshot of the field. So I think an early definition that I think still captures a lot of the spirit of synthetic biology today is really about the design and construction of new biological parts, devices, and systems, and the redesign of existing natural biological systems for useful purposes. So if we just look at the image and the text on this slide for a minute, you can see that there's a juxtaposition here of bacteria and gears. You can see terms like design and construction. So language and images like this sort of really start to give us a sense of the engineering-driven concern of synthetic biology. There's a desire to make living organisms, to repurpose living organisms, to make them behave more like machines so that they can ideally do useful things for us. And early in the development of this field of synthetic biology in the kind of early 2000s, we saw this kind of wave of papers being published that were really trying to discuss and frame this relationship between engineering and biology. And some framed this as a partnership between them. Others placed engineering kind of as the dominant discipline. Doesn't, regardless of your kind of take on exactly what the nature of this relationship is, over the past 20 years, there has definitely been an influx of engineers into the world of molecular biology. And along with this, they've been bringing engineering ideas and practices to bear when they're working with microorganisms, yeast, bacteria, and so on, and the kind of genetic makeup of these living things. And this sort of increasingly design and construction-based approach to genetic engineering has been the subject of a whole bunch of policy reports. I mean, if you look even just in Europe, the US and Australia, you're looking at like 60 plus reports over the last 20 years. And in these reports, what we see, there's lots of different things you can look for in them, but just to highlight today, this sort of sense that synthetic biology is being identified as an essential technology to meet grand societal challenges, especially around health, environment, climate, and so on. So it's really being positioned as having this critical role. And I would say another interesting and defining feature of this field, this kind of academic field from the outset, has been its emphasis on responsible research and innovation, and on including researchers from a wide range of social science and humanities disciplines. So there have been many different attempts to try and integrate social dimensions of synthetic biology quite upstream in the development of the field. And I would say these have often been top-down initiatives by research funders. So the National Science Foundation, the European Commission, and they've used kind of headings along the lines of responsible research and innovation, or human practices in the context of synthetic biology, as well as a more traditional label of ELSI research, where ELSI stands for ethical, legal, social issues. This was something that was pioneered with the Human Genome Project. And all of these top-down kind of funding initiatives are grounded in an acknowledgement that if we start to make serious improvements in our ability to engineer living things, this should also be accompanied by careful study of what these developments might mean for society. And I would say in the context of synthetic biology, the aspirations have been a little bit more ambitious than just contextualizing scientific developments. They've actually been concerned with trying to promote transformation of the scientific research process itself. So in various ways, they've been trying to integrate social scientific and humanistic inquiry within the more technical research process. I would say there's varying degrees of success. We can have a huge conversation about this if anybody's interested in sort of trying to understand what that has looked like over the past 15 years. But back in 2007, when I was first dipping a toe into this world of social science, I became part of this wave of, hangers-on, I guess, who were getting involved in synthetic biology through these types of initiatives. And since then, I've ended up basically participating in the growth of the field in lots of different ways. So I've been on research grants. Sometimes these have been standalone sort of STS projects, but more often they've been larger technically driven projects where I've been doing a kind of social scientific component of them. I've done a lot of teaching of synthetic biology students through both undergraduate and graduate courses, like full standalone courses in the UK, as well as here at ASU. And then I've also been part of all kinds of community building efforts for this field. So for example, chairing working groups as part of initiatives like the global iGEM competition, which is a huge undergraduate or a huge student competition. It goes beyond undergraduates now. And also the US's Engineering Biology Research Consortium. And I've organized lots and lots of meetings and workshops and conferences for the research community. So in that sense, you could say I've been very involved in, and some might even say quite complicit in the making of this field that comes with its own challenges. But I've had the chance to see and experience a whole variety of these experiments that have been undertaken with the aim of promoting responsible innovation. And just kind of, again, really high level take on this. I mean, it's more nuanced, but just to sort of, my sort of overall feeling walking away from these is that these experiments often fall short of really transforming the research process. They often end up feeling more like add-ons, or they feel like kind of extra work for everybody that doesn't really result in any clear benefits for anybody. And in practice, I think often these initiatives, although they might be kind of very well intended from, the funder's perspective, what they often end up doing is allowing scientists and engineers to actually detach themselves from the social and political dimensions of their work rather than really embroiling themselves in the issues. And to kind of give a couple of examples there, I think, especially with these highly interdisciplinary projects, there often ends up being a division of labor that ends up putting the responsibility for responsible innovation into the hands of the social scientists rather than the technology developers. And then also when it comes to the kinds of outcomes of these responsible innovation efforts, it feels very challenging to disrupt the predominant risk-based framings of what's at stake and to encourage a broader set of values to come into conversation. And I guess by that, what I mean is that it often feels like the only acceptable reason for questioning a particular technological trajectory is to say that there's a risk to physical safety. And that's kind of the only grounds upon which it's okay to consider other options, rather than acknowledging the possibility of concerns that might be grounded in quite different rationales. So this is sort of I guess this kind of pervasive framing that we keep bumping up against. So I would say 15 years down the line, even with quite serious efforts to foster these sort of more complex entanglements between synthetic biologists and the worlds that they're creating, I still think there are a host of conceptual and practical challenges. So it's not all doom and gloom, but it's sort of still have this sort of base level of sort of frustration and challenge around how we do this and how we do it well. So I'm really interested in thinking through this puzzle and thinking through ways that we might actually be able to kind of really push for a reframing of the relationship between synthetic biologists and their work. And so lately I've been thinking a lot about the idea of care as being a slightly different entry point into studying and intervening in this world. And I'll come back to care in a few minutes, but I thought I should introduce the case study for the work that I'm doing here, which is the Biofoundry or the biofabrication facility. These biofoundries are a new type of platform infrastructure that are being developed to enable kind of really high throughput genetic engineering. Again, mostly this is microorganisms. We're talking about yeast and bacteria and so on. And the idea of a biofoundry is ever so slowly making its way into public discourse. I think it appeared in the New York Times for the first time last week. So this word is starting to be, to get out there a little bit. But biofoundries typically involve investing in robotic equipment, like you can see in these images, and trying to design these integrated workflows around them that really configure the machines, software, databases, DNA, microorganisms, the humans working on them, sort of trying to create a new configuration of these elements to generate new forms of value. There's sort of a sense that there's something, if we can extract, there's a lot of value to be extracted from learning how to design organisms effectively. So the aim of these biofoundries is to develop standardized and ideally automated, this question of how much you can automate is an interesting one, but to develop these kind of standardized automated processes to reliably and efficiently design new useful organisms. And we see that these workflows, as you can see on this slide here, they're often framed in the language of an engineering design cycle. This is where you sort of see these engineering ideas pushing their way into biological practice. Iterative cycles of designing, building, testing, and learning, and sort of trying to configure these workflows in productive ways. And so over the past decade, universities and startup companies have been setting up biofoundries thanks to tens of billions of dollars of investment from a variety of public and private sources. So we've got academic biofoundries and then a whole set of companies that are also emerging in this space, that startup companies, but also increasingly in the last few years, a few companies that have gone public. So each of these biofoundries, I see them essentially as their own design project. They're each trying to figure out their own efficient and effective ways of reconfiguring life at the genetic level so as to extract or create new sources of value. And in practice, this is proving to be a very challenging project for them. It's not straightforward to figure out what a biofoundry looks like or should look like. These practitioners, from my observation so far, they are engaged in sort of a continual sense-making process. They're constantly, very self-consciously reconfiguring how all the components of their facilities fit together in pursuit of this imagined value down the road. And so with the number of different biofoundries that are currently under development, you could say there are lots of parallel experiments underway, which makes for really kind of interesting and fruitful comparisons at this point. So again, just to give you a little flavor, some of my early visits to biofoundries have revealed very visible differences across just the physical configurations of different sites, different physical layout of equipment. But I would say, this introduces different imaginations of what the future of work looks like. They suggest these different configurations suggest different strategies for how humans should engage with machines in doing biological design. So, for example, this first picture here is from the Concordia Genome Foundry in Canada. They have a model where they have these individual separate workstations, each one of which is oriented around different equipment and capabilities. If you look at this picture, you might not actually really see any big difference between this and a normal lab. Some biofoundries have taken this kind of individual workstations model to a kind of a different level. The Agile Biofoundry, for example, is distributed across seven different national labs. So it's not just distributed like within a lab, it's across different sites. But on the opposite side of the spectrum, you see some of these biofoundries that are aiming for sort of total automation, and they have these fully integrated systems where robotic arms are moving samples between one piece of equipment and the next. So in principle, you just kind of introduce some samples. They don't usually get them out of the fridge for you, but you introduce some samples into the system and you walk away and the design happens. And then there are also these kind of curious examples of hybrid approaches. So for example, the company Zymergen designed these special interlocking carts that literally allow you to like reconfigure your workflows and snap the carts together to carry out different processes. And I would say these are all, examples of different imaginations of what human work or labor in a biofoundry should look like. And we also see this in the self-presentations by these different facilities. So again, if you go to their websites, you can see that these facilities make very different choices about whether to highlight or downplay the role of humans in their work, right? So different visions of how much we're relying on automation and some sort of snazzy, sterile, kind of vision of the future, or whether it's, full of messy humans working with messy biological organisms. So in this sense, I see the existing biofoundry or this emerging biofoundry landscape as a site of very active experimenting. Each facility is really actively trying to figure out how to build a successful platform based on their vision of the future, what they think is important to do. But I think the question of what success means is still very much up in the air. So for most commercial biofoundries, success, at least to some degree, is tied up with being able to create value through processes and products, right? They have to somehow make money in order to survive. For the academic biofoundries, the emphasis is probably a little bit less oriented towards making money, but there are still big questions about how to develop a sustainable business model, and that is far from clear at this point. But I would say also for many biofoundries, there are additional layers, for other companies too, but for these foundries, there are additional layers to this question of what success means, because the visions of synthetic biology that are circulating out there go way beyond simple economic goals. They include much bigger visions for social transformation. So if we go back to some of the policy reports that I mentioned earlier, synthetic biology is presented as an approach that will make, significant contributions to environment, energy, health problems. It's touted as being a huge driver for a more sustainable bio-based economy. Rhetorically, there's a sense that this technology could be associated with a whole set of very positive interventions in the world. So in that sense, too, the success of biofoundries is, at least to some extent, bound up with its ability to make these not just money-making, but kind of positive interventions in the world, while also minimizing, potential negative impacts, misuse, and so on of these platforms. Which I think brings us back to these kind of thorny questions around responsible innovation. So, how do these practitioners in these sites innovate successfully and responsibly? And, for me personally, like, what does my work as a researcher and a practitioner look like when kind of engaging with these facilities? And I think, responsible innovation in the context of these biofoundries is particularly important because they are infrastructure building activities. And infrastructure building is kind of a world building activity, right? The aspirations of these foundries are literally being given shape through the infrastructure that they're designing. And like with any infrastructure projects, there are tons of design choices in play right now. And these choices we know can end up shaping the sorts of questions that we can ask and the kinds of outcomes we can expect. So, we can ask about the kinds of futures that are being promoted and the kinds of futures that are not being tended to as this infrastructure is being developed. So in my research, I'm really interested in studying the kinds of values and design choices that are being built into this infrastructure, as well as trying to create opportunities for these practitioners in these facilities to reflect on and grapple with their choices and to think about how they can sort of, turn those reflections into action. And as I mentioned, I'm sort of doing this work as part of a project that frames responsible innovation through a lens of care. And I'm using care kind of as a theoretical lens because of what it draws our attention to. So I think a concept like responsible innovation can get quite disconnected from the day-to-day actions of people or of organizations. And I think it kind of often falsely leads us into this dichotomy of assuming that we're either behaving responsibly or irresponsibly with like not a lot of room in between. But in contrast, the concept of care gives us a lot more nuance and wiggle room to play with. So, I mean, you can even just start by asking, like, what is it that we care about? And you can ask this of individuals and of institutions, right? And we'll probably find that a whole bunch of different values start to emerge when we ask this question of, what do you care about? And some of these might align well with each other, and some might be in tension with each other. So, I might simultaneously care about making money and having a meaningful job that I, derive some pleasure from. And those things I might be able to align quite nicely. I might also really care deeply about sustainability. It might not be quite as clear whether making money and being sustainable are quite as neatly aligned as I would want them to be, right? But I can start to ask questions then, like if I really want to align these two things, if I really want to both make money and do work that contributes to sustainability, are there specific things I can do? Are there actions I can take to make this happen? I can think about this as an individual, but we can also zoom out to the level of the institution and say, are there ways of designing this infrastructure to try and promote more alignment across the many different things that we might care about? So this new research project that I'm working on involves studying biofoundries from sort of these multiple different vantage points. And so I'm spending time following the everyday, often mundane practices of care that are underway in these biofoundries and looking at how these everyday practices are helping to build this new kind of infrastructure and how this translates into the details of the infrastructure that's being built. So here I'm talking about paying attention to the detailed, the jobs people are doing, their personal stories, their aspirations for their careers and their lives, and also the types of dynamics that are at play within teams, across different teams, and so on. And I think understanding this everyday work of care is really important because these seemingly routine tasks like managing a team or maintaining your equipment or curating your data, they're all critical to success, to what a successful biofoundry will be. And so looking at where and how people are investing their time and their effort helps us to see the kinds of values that are being built into the infrastructure and helps us to see how biotechnology is being reconfigured through the work that these facilities are trying to do. But we can't get the whole picture just by looking at what's happening inside the foundries. We also, I think, need to zoom out and start thinking about the bigger political economy in which foundries are taking shape. So we need to pay attention to the kinds of resources that are being invested in these facilities, the regulatory frameworks that shape what's possible, and the broader imaginaries that have allowed these foundries to emerge as an innovation in the first place. And we need to do this to, we need to understand the different incentives and constraints that are operating on the institution in order to understand the kind of landscape, the possibilities for innovation. And I think what's interesting here too is not just to focus on kind of individual kind of practices and moments here, but to look for synergies and tensions in the data that you're collecting. So places where the practices and the politics of care seem to be aligned well, or where they're at odds with each other. And then the idea in this project is to sort of use these synergies and tensions to reflect back to practitioners what's going on and to create opportunities for dialogue and potential change in the name of responsible innovation. And so doing this kind of project, I think, really clearly depends on having the participation of biofoundries. And I'll say that so far, interestingly, I found a lot of openness among these foundries to engage with these ideas. So for example, last week I joined probably the biggest biofoundry in the US for a tech chat that was all about care. This is a company that's really actively trying to grapple with what it might mean to care for your platform. They've been looking hard at Silicon Valley and at the tech industry and saying, we don't necessarily want to fall down those same traps of, say, oh, we're just platform designers. We don't care how our platform is used. And so they're really trying to think about, well, how do you do that? And so they're having really challenging conversations right now about how you sort of integrate these multiple competing demands to make decisions that you feel as a company you're happy to stand by. So, I sort of painted this kind of doom and gloom earlier about, I just really don't know how far we're getting with this whole responsible innovation thing. But it might be that, 20 years into the making of this field, we have, within synthetic biology, a group of practitioners that have, at least to some extent, really internalized responsible innovation as being part of their efforts, but are trying to figure out how to do this. And I think it may also be that, we're in a particular moment that has brought increased visibility and urgency to questions around care and justice and equity. But I think, as a site, these foundries are proving to be very interesting to sort of focus on, to see, really to test the extent to which you can kind of, apply responsible innovation in these upstream settings. So I'll leave it at that. Thank you all so much for your time this morning. And if we have time for questions, I'd love to engage with you.
Thank you so much, Emma. That was brilliant. We've been spoiled today, everyone, from both Rebecca and Reese, and now Emma. Questions for Emma? I'm sure there are many, actually.
I do have a question for Emma, if you don't mind. So would you consider DNA editing, like with CRISPR, is that fall under your realm of view?
Yes, it does. Interestingly, you could argue that the sort of labs that CRISPR came out of they don't necessarily call themselves synthetic biology labs, but the synthetic biology community has been incredibly quick to adopt this as a success story and as sort of evidence of the potential of kind of gene editing, genome engineering.
So care takes some significance there, right? I mean, there's so many different issues involved with that. So very fascinated. Thank you.
Emma, could you comment on what care does to productivity?
Yeah, it's a challenge. And this is actually something we were talking, with this company last week, kind of talking about too, right? Is sort of, does it slow things down? Does it change the kinds of, does it make you ask different sorts of questions before you take on a project? Does it make, what are the things that you should attend to before making decisions about, kind of how you're going to do things, like the sort of mantra of like, move fast and break things, doesn't necessarily sit easily alongside. But at the same time, I think there are ways, it depends on how you think of care. And to some extent, if you think of care as an ongoing commitment, where you say like, I am committed to standing by the work that we're doing and to the effects that it has in the world, you might not say it has to like dramatically affect productivity to start with. But what it does mean is that you are responsible, you take ownership and responsibility for what happens to your products out in the world as well. So if you want to move fast and just put things out there, that might be fine. But that you are committed to, any cleanup or any mess that might ensue rather than backing away. So I think it's a really complicated, it's not a straightforward relationship with productivity, because you could, sort of, if you stand by it, you might say, okay, I'm going to do this more slowly and, feel more confident about what I'm putting out there. Or you might say, no, we're just going to do it. And, and then if there are consequences down the line, we'll deal with them.
What a great moment to finish on. And Emma, that is just so interesting. So interesting. I know we had done early studies looking at implantable devices, for example, in the 2000s, and we came up with the 4 C's. It was control, care, convenience, and somebody told us we had to put in the cool factor, and then to look at the dynamics between these 4 C's. but we built an umbrella kind of diagram, which was the one that was the control, and everything else, no matter what, was underlying. So there was control at the top, and then sort of care, convenience, and cool beneath the umbrella. But some of that work was interesting. I think it relates to some of the biofoundry work. I mean, some of the biofoundry people are transhumanists in some of the smaller laboratories, but your work we have to watch over the next 10 years, particularly 5 where you've been funded by the NSF to do this work. And I think you're going to be one of these leading voices, actually asking those significant questions with care, might I add, excuse the pun, because that's your work, and that's what you delve into. So we wish you every success. And to Rebecca Herold and Reese Farthing, to you two on your missions and your advocacy roles, we can't wait to see where you'll get to. So next week we have Martine Perez presenting. It's our last one for the whole year. And I just want to thank all the diehards for coming each week and listening to our guests. Bye for now.